Hello, everyone. This will be a combination of chapter 3 and 4 lecture, and this is more of a basic and a brief overview. I have other videos that have more in detail instructions on how to do things that you will be learning this week. For example, how to do a Punnett square and how to analyze a pedigree. And so I do have videos with more in-depth detail, so I will be doing just a brief overview of chapter 3 and 4, which I have combined for this week. Just remember that next week you will have your exam, and I won't be assigning any more chapters next week. I will just give you the exam only. So I decided to combine, instead of breaking up chapter 3 and 4 into 2 weeks, I decided to combine them into this week since you'll need a little bit more time to study some of these concepts. So we'll start off learning about Gregor Mendel. He is considered the founding father of genetics. He was an Augustinian monk, earned his physics degree, and then became a teacher. And in 1854, he began his experiments in genetics. Gregor Mendel used pea plants to study genetics. And what's great about pea plants is that there's a lot of identifiable traits. For example, flower color and pea size, pea shape, pod size, pod color. All of those traits is something that you can easily see, and so those were very clear phenotypes. And based on that, they were easily traceable as well. And what's another nice thing about pea plants is that they have a short generation time, and so they can reproduce in a few amount of weeks, and therefore he can record what goes on and how traits are carried on from generation to generation. And so studying genetics is not that easy, let's say, for example, in humans, because humans have some easily identifiable traits and others that are not so easily identifiable. But, studying family lines, studying generation from generation can take hundreds of years. And so it's just not something that you could easily do. Um, all right, so some of the traits that he studied with his pea plants were shape, color, pea coat color, and this was specifically in peas. He also looked at the pods, the shape and color, the flower position, and the stem length. And some of the traits that he noticed, and so this was after doing his experiments, were dominant. And so he noticed that the smooth characteristic of the peas were dominant, and wrinkled is recessive. And again, we'll go over that in a little bit. And so he noticed all of these different characteristics about pea plants, which are dominant and recessive. Dominant means that that phenotype or that trait occurs more often, and as long as the organism has one genetic trait or one gene for that trait, it will be expressed. So one of the first experiments that he did was basically take a look at the trait of smooth versus wrinkled phenotype. And so this smooth phenotype can be found in peas and also the wrinkled peas. And what he did was he took a generation, he called it P1, so this is the first generation, the parental generation. And he took a female plant and crossed it with a male plant that had these very specific traits. And he wanted to know what the next generation would inherit. Would the next generation inherit a combination of smooth and wrinkled? Would they all be smooth? Would they all be wrinkled? Or would it be 50-50? And so when he crossed these two parents, he found that the first generation, called F1, basically all of these plants had smooth peas. And so even though one of the parents had a wrinkled phenotype, it was not passed on to the next generation. All of the plants had smooth peas. And so he was curious. He wanted to know, okay, where did this trait go? Why was this trait lost? And so what he did was he took two plants from the next generation, from the F1 generation, and those two plants we know both have the phenotype of smooth peas. He crossed them, he self-fertilized them, and he found that the F2 generation, so this is a generation after the F1, and so therefore the parents were gathered from the F1. The F2 generation had an interesting characteristic. He noticed that 3/4 of the plants, and so if you notice here, there's 5,474 plants, so that's a lot of plants, it's a lot of data. He noticed that 3/4 of those plants had a smooth phenotype, and that 1/4 of those plants had wrinkled peas. And so this was interesting to him because what he found out through this, what he figured out was that traits are not necessarily lost. You can carry a trait and not express it, and that trait can then be carried on to the next generation. And so here's another example. Let's say you have two types of flowers. You have a parental flower. Let's say these are females that are purple and males that are white. And so you can cross these two flowers, and the F1 generation, in this case, would have all purple plants. And so what happened to the white flower phenotype? So where did that go? So if you take two of these plants from the F1 generation and cross them again, you can recover the white flower phenotype, and also get the ratio of 3/4 having purple and 1/4 of them having white. And so he did this again with a lot of different traits. He did this with yellow versus green peas, gray versus white, full pods versus constricted pods, green versus yellow, and also the flower orientation and long stems versus short stems. And he kept getting this ratio of 3 out of 4 plants would express what we call now the dominant phenotype, which is the phenotype that is expressed the most, and 1/4 would continuously express the recessive phenotype. And so the trait wasn't necessarily lost, it was just expressed in later generations. And so what he found out by doing these experiments is that traits, he coined them, he called them genes, are the fundamental unit of heredity. And that unit can be passed on to later generations. And he also discovered that some of these genes are dominant, meaning that as long as the organism or the offspring, so the offspring are the children, so as long as the children have at least one dominant gene, because they can inherit too, as long as they have one dominant gene, that gene will be expressed. That will win over the recessive. So it'll mask the phenotype of the recessive. And the recessive gene is the one that is not expressed unless the organism inherits two recessive genes, one from the mom and one from the dad. And from this, he also discovered that each pea plant carries two genes for every given trait, one from the mother and one from the father. And then he coined those traits in a specific way. He said that the observable traits, for example, how you can observe smooth and wrinkled, he called that a phenotype. And so that's something that you can actually see. And the genotype is the actual genetic code. And so what is the code? What is the gene that encodes for this type of phenotype, for the smooth and the wrinkle? So that genetic code is in the DNA. And so that's a genotype. And he also found that every given trait, for example, like the peas, they can have a variation of alleles. So peas can be wrinkled, peas can be smooth, peas can be yellow, peas can be green. And so alleles are variations of a given gene. And so another example of that, where we see a lot of different alleles of a given gene, are eye color. And so there's a lot of different types of eye color. And this is just one example. There's, of course, we have many, many genes with a lot of alleles, okay? A lot of variations of that gene. And so going back to eye color, there's blue eye color, hazel, green, any, variation in eye color is basically a different allele for that gene, for eye color specifically. From Mendel's discoveries, scientists figured out that genes are found on chromosomes. And so we know that genes are inherited in pairs, one from mom, one from dad. And they also segregate when they're passed on. So the offspring doesn't necessarily inherit both of those traits from one parent. It can only inherit one trait from one parent and another trait from another parent. And that's similar to chromosomes. So chromosomes, when they're passed on, only one chromosome can be passed on to the offspring, not both. And so, for example, I have my chromosomes because I have one set for my mom and the other set for my dad. And so that means they each can only pass on one trait to me. And so the similarities in how genes are inherited and how chromosomes are inherited led us to surmise or let scientists to figure out that genes are actually located on chromosomes. And genes are also found in a very specific location, and that location is called a locus. And so a locus is a specific location in which the gene is found. And so this gene, this red one, let's call the red one the not the gene, sorry, this is called the red one a chromosome. So this is a chromosome that you inherited from your mother, and this is a chromosome that you inherited from your father. And so the gene location, so this capital P here, in the chromosome that you inherited from your mother is found in this very specific location, this very specific loci. And it's also found in the exact same location on the chromosome that you inherited from your father. And so that you see it right here. So this capital P, capital P. And by convention, a dominant phenotype is expressed in capital letters. And so let's learn some other quick terms. So homozygous is basically the inheritance of two of the same type of genes. So for example, if you inherited two dominants, or if you inherited two recessives, and so recessives are written in lower cases, that's considered homozygous. So you either are inheriting two dominants or two recessives. If you inherit a dominant and a recessive, that's called heterozygous, meaning that there's a mixture of the genes, and so one is dominant and one is recessive. By convention, as long as you inherit one dominant, so you can be two dominants or you can be one dominant, as long as you inherit one, you will always express that dominant trait. The only way that you can express a recessive trait is if you've gained a recessive trait from your mom and from your dad. So just going over again homozygous and heterozygous. So let's say there is a smooth phenotype, and a plant inherited either the capital SS, so this means the dominance for the smooth phenotype. And so if they are homozygous for the dominant trait, it would be written capital S, then that plant will express a smooth phenotype. If a plant inherits a recessive, so this is little s, that means that it's recessive, and the recessive trait in this case is wrinkled. So if there's no dominant gene, then the recessive gene will be expressed. And so the recessive in this case is wrinkled. And this plant here inherited from its mom one recessive gene, and from the dad another recessive gene. So this is homozygous recessive. And now this plant expresses wrinkled peas. Now, if these two plants were crossed, so let's say we cross a dominant homozygous plant with a recessive homozygous plant, let's say we cross them, then the F1 generation, the next generation, will have one capital S from, let's say, mom, and one small s, let's say, from dad. And in this case will be heterozygous, carrying one dominant trait and one recessive trait. But even though it's heterozygous, as long as the dominant gene is there, that dominant gene will be expressed, which is smooth. So by convention, dominant genes are written in capital letters, and recessive genes are written in lowercase letters. So make sure you know this, especially when you're making a Punnett square. And so a Punnett square is a neat way to determine what the phenotypes and genotypes of the offspring may be. And so this is based on probabilities. So these are the odds of the offspring coming out this way, meaning that this doesn't always happen, but the odds are very high. And so let's say we wanted to figure out what the phenotypes and genotypes of the parents with heterozygous genes are. So let's say we have one parent which is heterozygous for smooth, meaning they have one dominant and one recessive trait, and the other parent that is heterozygous for smooth again, which means they have one dominant and one recessive. Let's say we cross them. Well, the way that we can figure out what the odds are for their offspring to have certain types of phenotypes is by doing a Punnett square. And so this here is a Punnett square. You basically draw a box with four little boxes on the inside, and you put one parent on the top. And so in this case, it's the male, so capital S, little s. And on the bottom, it's the female, so capital S, little s. And then all you do is cross them. So let's take a look down here for the cross. And so here we have one gene that is passed on from the female, which is a capital S, and the other gene which is passed on by the male, which is a capital S. And so now this offspring here, this child of, of these pea plants is smooth and has inherited two dominant S's. And the next child is a capital S and a little s. So here's the little s, here's the capital S. And the other offspring is another capital S and a little s. And the last offspring here is a little s and a little s. So based on the inheritance of two recessive genes, now this plant here is expressed as wrinkled. So that means that this plant has wrinkled peas. However, the other three plants, just because they have one dominant gene present, so for example, this one here has one dominant gene and this one has one dominant gene, the dominant gene is the one that's being expressed, and so that's smooth. And so if we look at the phenotypes, 3 out of 4 are smooth and 1 out of 4 are wrinkled. If we look at the genotype, we know specifically, so this is the type of genes that it has, we know that one plant has two dominant genes, and we know that two plants are heterozygous, so that means that they have one dominant and one recessive, so therefore they are a carrier of the recessive gene. And when you're a carrier, you have the potential of passing it on to your next generations. And there is one offspring that is recessive, homozygous recessive, for two wrinkled phenotypes. When you make a genetic cross, this is also called a Punnett square. And so a Punnett square is basically like a square. So you draw a square with 4 squares on the inside, and you put the genotype on top. And so it can be from the sperm or from the egg. It doesn't matter which one goes first. And so you want to put one allele on one end and the other allele on the other end. And so in this case, both parents are heterozygous for this gene called A. And so this gene is irrelevant, but it's called A. And so this is heterozygous for A, and this is also heterozygous for A. And when you cross them, you get the same type of cross that we saw earlier, which is a homozygous dominant, a heterozygous dominant expression, heterozygous dominant expression, and homozygous recessive expression. And so this is the only one that'll express a different type of phenotype, simply because the offspring inherited two recessives. And so let's take a look at the Punnett square of these purple flowers and these white flowers. And so let's say we, in the P generation, the parental generation, we take one parent that is dominant, so this is homozygous dominant for purple, and we take the other parent that is homozygous recessive for white. Let's cross them, and when we cross them, we notice that all plants have the purple phenotype. So remember, this is what you can visually see. However, the genotype we know is heterozygous. So we know that the genotype is one capital P and one lowercase p. So now if we want to figure out what the F2 generation will look like based on crossing two plants from the F1 generation, then we can make a Punnett square. So I would suggest you make a Punnett square with the parents having a capital P and a little p. So both parents have that gene. So go ahead and make a Punnett square. You can pause this and see if you do it correctly, and then come back to the video and check your work. So if you did your work correctly, what you would do is you would write capital P and lowercase p on the top, so that's one parent, and capital P and lowercase p on the side, that's another parent, so this can be male and this can be female. You would draw a square, and then you would simply cross. You would get two capital P's, so this is a homozygous dominant. Then you would get a heterozygous dominant, and so this is capital P, little p, although the dominant trait is being expressed. And you would also get another heterozygous dominant, and then you would get a homozygous recessive. And so there would be 3 out of 4 plants that are expressing the purple phenotype, one being homozygous dominant, 2 being heterozygous dominant, and you would get one plant expressing the white phenotype, which means that they are homozygous recessive. Now, not all genes follow this rule, although a lot of them do. which is what Mendel discovered, but not all of them do. And so there's a case in which there can be incomplete dominance, meaning that both genes are kind of like a mixture of each other. And so in this case, for example, with incomplete dominance, you have a red phenotype expressed in capital R, and so this parent here has RR, so it's homozygous dominant for red, and the other parent here is homozygous recessive for white, and so it's written in the lowercase white. And when you cross them, you end up with an F2 generation of heterozygous dominant, but even though it's heterozygous dominant, it's a mixture of the previous genes. So in this case, it's pink. And so although it's heterozygous dominant, the color is pink because there's incomplete dominance, meaning that one gene is not dominating over the other. They're both like a mixture of each other. Now, when you do Punnett squares, and when I have you do problems in class, unless I mention that there's incomplete dominance going on, don't assume that it's incomplete dominance. Assume what we talked about before, the rules that the dominant will always be expressed, and when there's no dominant around, then the recessives will be expressed. So we'll assume those rules unless I specifically tell you there's incomplete dominance or some other type of dominance going on. So now there's another type of gene expression called codominance, in which both of the dominant traits are expressed. And for example, we find that in our blood type. And so we have these glycoproteins in our blood, and some are called M and some are called N. However, if you inherit the M glycoprotein and the N glycoprotein, both of those proteins will be expressed. So you don't see one dominating over the other. The M won't dominate over the N, the N won't dominate over the M. Both glycoproteins will be expressed on our blood cells. So that's considered codominance. Sometimes multiple alleles are expressed as well, and so you don't have like just one variation or the other. There can be many different types of variations, and one example is in our blood type. And so some people have the blood type A, while others have a blood type B. And so basically those A and B are molecules that are basically like identity tags recognized by the immune system. And so again, some blood cells can have can be blood type A, some can be blood type B, some can be a combination of both, and so that's like codominance. So you have A and B, or you can have the complete absence of either of those proteins. So it can just be blood type O, so not A or not B. And so if you notice, these are multiple alleles that can be expressed within a population, just like eye color. All right, so let's go on to the next thing, which are pedigrees. And pedigrees are used to analyze human traits. And this is very useful to find out how a genetic disease was inherited. And so you can go back in your family line, so you can go back in time and look at your family line and also how certain diseases have been inherited. And you can also make predictions on how future generations will be affected. And people like to know these things so that they can make informed decisions if they are deciding to have children. And so maybe their partner has the same type of genetic disease that they have. And so you might want to consider the risk of maybe having children, if there's any risk at all. And so a pedigree is basically used to determine whether the trait has a dominant or recessive pattern of inheritance. And some diseases don't necessarily come from just autosomes. Some diseases are specifically located on an X or a Y chromosome, and so the X or the Y chromosomes determine our sex. And that's called an X-linked disease or a Y-linked disease. And so you can use pedigrees to also determine whether that disease is X-linked or Y-linked, because that kind of changes the rules or the patterns of inheritance when it's X-linked or when it's Y-linked. So a pedigree is basically drawn in squares and circles, and so squares are males, circles are female. The line that is formed across is considered a mating, And the line that is formed down, like perpendicular, is basically the next generation. And so the I here is generation 1. The 2 is generation 2, meaning that the female and the male have mated and they had 3 children, 2 males and 1 female. And another important thing for you to know with these pedigrees, at least for our purpose and for this class's sake, is that when there's a gene or like a disease of interest and you want to follow the pattern, then the affected individual is basically shaded. And so we use a shading color which represents the individual which has either the trait, the characteristic, or the disease, depending on what you're looking at. And so this is what a pedigree looks like. And so, for example, this means that the male has either the disease or the trait of interest or whatever it is that you're trying to follow. And so that's why the male is shaded in. So this person right here is carrying that trait. And since this person here, the female, is not shaded, that means that the female is not carrying the trait. And so when the male and the female mate and they have offspring, the next generation does not have any children that are expressing the trait that was expressed previously. But it doesn't mean that these are not carriers, so they can be carriers of that gene. And so in this case, the female was a carrier. And when she mated with the male, even though he's not a carrier, her children, one of the males, had or inherited the trait. And so, as you can see, traits can skip generations, but it doesn't mean that the person is not carrying that gene. It can just skip a generation, and the next generation can express that gene. And so you can do this with your family line. You can go as far back as possible, and the farther back that you go, the more of a clear idea that you have about that type of gene. It'll let you know whether it's a dominant type of gene, a recessive type of gene, if it's autosomal, or if it's X-linked or Y-linked. All right, so autosomes and sex chromosomes. So all the chromosomes that are not sex chromosomes are basically autosomes. And sex chromosomes are the X and Y. And so the X and X is for females, and X and Y is for males. Even though these chromosomes are considered the sex chromosomes, it doesn't mean that all of the genes that are on those chromosomes determine the sex. So there's plenty of genes that are on there, quite a bit of them, that are unrelated to whether the individual will be female or male. For example, color blindness is one of them. And it tends to occur more in males than in females just because of the way that traits are passed on. And so a male has an X and Y chromosome, and a male will always pass on either the X chromosome, so it has a 50% chance of passing on the X chromosome, and a 50% chance passing on the Y chromosome. And so the Y chromosome is always gained from the father. And the X chromosome, in this case, is gained from the mother in the males. And so if the mother is carrying, let's say, 2 recessive phenotypes, let's say of a disease, then the male will always express it because it'll always gain 1 recessive from the mom. And so you see that the patterns of inheritance change when it comes to the diseases being located on the X and Y chromosomes. And let's say that disease, again, the female has 2 recessives, but let's say the dad has a dominant. Well, the females will never gain the disease because they will always gain a dominant gene from the dad. And so females in this case are spared, but not the males. And so that's why there's an increase in the color blindness disease in males, just because they can inherit it more easily. And so there are 5 basic patterns of Mendelian inheritance, and that is called autosomal recessive, which is basically a characteristic that is inherited through recessive traits, meaning that you have to have 2 recessive traits in order to express it. And these are on the autosome genes, so that means that autosome chromosomes. So that means that this is found on the chromosomes that are not related to the sex chromosomes. Autosomal dominant means that as long as you have 1 dominant gene, as long as you inherited 1 dominant gene, you will express it. And that characteristic is found on all the chromosomes, or I mean, not all, right? But on one of the chromosomes that is not related to the sex chromosomes. And an X-linked dominant means that it is found on the sex chromosomes and that this disease is a dominant uh, disease, and so it takes a dominant gene to express it. And if it's X-linked recessive, it means that it takes a recessive gene to express it, but yet it's found on the sex chromosomes. And if it's Y-linked, well, that means that this disease is related only to males, because only males can inherit a Y chromosome. So here are some examples of autosomal dominant traits. So you can just take a look at this list, and some of the ones that we have talked about are Huntington. Maybe some people know or have family members that have Huntington disease, and so this is expressed in the later stages of adulthood. There's Marfan syndrome, and your textbook talks about Marfan syndrome. I believe Abraham Lincoln had it, and so that's what made him unusually tall and kind of long, because of this loose connective tissue defect. And there's nail patella syndrome, meaning that the individual is born with the absence of nails and kneecaps. And so these are dominant traits, meaning that as long as one dominant gene is inherited, the individual will express that disease. But although a disease is dominant, it doesn't mean that it is in high frequency in a population. A disease can be dominant, but it doesn't mean that the majority of the population has it. So that's not true about dominant genes. Autosomal dominant genes basically have a 50% chance that the child will inherit it. And so if an individual, for example, the female is carrying that dominant dominant gene for the disease, and it also depends whether she's homozygous dominant or heterozygous dominant, regardless, there's a really good chance that the offspring will express that gene or disease or that trait or characteristic. And it'll also be passed on to the next generations. And so just by looking at this pedigree, we can surmise, or we can assume here, that this characteristic is most likely a dominant trait. Now let's look at some recessive traits. And so these are recessive traits that are found on the chromosomes, which are not related to the sex chromosomes. So those are autosomal. And you've probably heard of albino, like albinism, excuse me. And so this is the absence of pigment in the skin, eyes, and hair. Some other types are sickle cell anemia, so we talked about that before. And this is the abnormal hemoglobin, which doesn't bind oxygen very well. We've talked about phenylketonuria and also Tay-Sachs disease. And so these are examples of recessive traits, meaning that you need to inherit a recessive trait from your mom and a recessive trait from your dad in order for that characteristic to be expressed, in order for that disease to be expressed. If you did not inherit the recessive from mom and dad, then you will not show that disease. So even if you have just one dominant gene that is not related to the disease, that dominant will mask the recessive. And so you will not express the disease because you have a dominant that masks it. But if you're a carrier, it means that you have the potential of passing it on to your children. An autosomal recessive gene has a 25% chance that the child will inherit it. And so if we take a look at a pedigree and we notice the parents, for example, do not express that trait, but they are carriers, one of them is a carrier, because the next generation has one offspring, here the female, which is expressing that trait. But notice that the parents had 1, 2, 3, 4 children, and so that means that 25% chance, 1 out of 4, that will express that gene. And so if they are expressing that gene and this is recessive, that means that both parents are carriers, because that's the only way that this person can express that gene. That means that they inherited one recessive from the mom and one recessive from the dad. And so this trait can be passed on to the next generation. So you see here that the male is a carrier. They did not express the gene, but they are a carrier. And that was passed on to the next generation, to his son. And so just based on this pedigree and based on the low frequency of this trait being passed on, this right here is a good example of what an autosomal recessive inheritance looks like. Then there's X-linked dominant traits. Like we said, X-linked dominant means that it just takes one dominant gene on the sex chromosomes in order for it to be expressed. And so all affected males, and so if your dad, let's say he's carrying the dominant gene, he will always pass that dominant gene to the daughter, but not to the sons, because the dad can only pass on the Y chromosome to the son, but will always pass on the X chromosome to the daughter. And so therefore the daughters will always be affected. That's if the dad is carrying a dominant gene. A heterozygous affected female, so this is a female that has one dominant and one recessive, will transmit the trait to half of her children. So that means that 50% chance that the daughter will carry that dominant trait, and 50% chance that the son will carry the dominant trait. So on average, in this case, this is because the dad is carrying the dominant. So on average, twice as many females are affected as males. So this is what a pedigree of an X-linked dominant trait looks like. And so in this case, the female here is dominant, and she basically passed it on to the male and female. So this is a 50% chance of passing it on, right? Because you have 1, 2, 3, 4 offspring. So that there's a chance that that means that this individual is heterozygous, because there's a 50 chance of passing it on, or else all of the males would have been affected. And so in this case here, where the male now is dominant, okay, so the male is dominant for that gene, since the females can only inherit an X from the dad, only one X, and as long as there's one dominant gene present, you will always express it. All females in this case are affected. And so just by looking at this pedigree and seeing how all females are expressing this trait, we can infer that this type of genetic disease here found in this pedigree is related to an X-linked dominant trait. Now, we can just kind of assume based on these pedigrees, and there's a good chance that I will show you these pedigrees, and I'll want you to figure out, okay, what kind of disease or what kind of pattern of inheritance do you see just based on this. But you wouldn't normally do this. You would want to look at many generations before you come to a complete conclusion. But just for the sake of our class, and just for the sake of these examples that you can find in your textbook and the ones that I'm showing you, I would want you to be able to tell just by looking at the pedigree what kind of pattern of inheritance is going on. And so make sure you take the time to look at these on your own if it's not too clear. The textbook gives a lot of information and examples, but all of these pictures that I have here from my PowerPoint come from your textbook. All right, let's look at X-linked recessive. And so X-linked recessive is basically the characteristic or the disease is inherited through a recessive trait. So that means that as a female, I have to have 2 recessive genes, one coming from my mom and one coming from my dad. But as a male, if the mom, let's say, is homozygous recessive, the males will always be affected. And nothing can come from the dad, because the dad does not pass on any X genes, or excuse me, X chromosomes to the son. The dad can only pass on a Y chromosome to the son. So this is all from the mom. And so X-linked recessive conditions are much more common in males than in females. Affected males who receive the mutant allele from their moms transmit it to all their daughters, but not to any of their sons, just like we talked about the cases earlier. And daughters of affected males are heterozygous and therefore affected, but sons of heterozygous females have a 50% chance. And so let's say that the female has one dominant and one recessive. There's a 50% chance that the son will be there's a 50% chance that the son will inherit that trait. So let's take a look at this pedigree in which the dad is expressing the recessive trait, and in this case the female is not. And so simply because the dad is expressing the recessive trait, that trait will be passed on to the females, but the females are not going to express it. They're just going to carry that gene because they gain that one X from dad and the other from mom. And remember, this is a recessive trait. And so unless you have 2 recessives, it will not be expressed. But all the females from this generation will be carriers, inevitably carriers. So even if they had 4 females or 5 females, they would all be a carrier of that gene. And when she passes on, so she has a 50% of passing on that gene to a male. And so in this case, you'll notice that the males are automatically affected because they only need to gain one X chromosome from the mom in order for them to express that recessive trait, since they only have one X. And so as long as they inherit just one X recessive from the mom, they will express that disease. And this male here is also expressing that disease. Same thing that's going on here. So if you notice, the male here is expressing the disease simply just because one of the X's was passed on with the recessive. And for males, since they only have one X, and this is X-linked, they only need one X to express that disease. Again, if this is a little bit confusing, just slow down, think about this, read this over again, and just follow the pedigree, because I feel like the pedigree is very self-explanatory. You just kind of have to think it through. All right, and so here are some X-linked diseases. For example, we talked about color blindness, and so males tend to have color blindness more than females. Hemophilia is another type of X-linked disease, meaning that this gene is found on the X chromosome of the sex chromosomes. And muscular dystrophy. Now, I'm not going to go over Y-linked genes, and I'm not going to go over mitochondrial related diseases. If you want to know more information about that, that is in your textbook, and it's pretty interesting. However, I'm not going to test you on that, so completely up to you if you want to learn more. And also, I know that I briefly went over the pedigrees and I briefly went over the Punnett scars, but I have other videos that go into everything way more in depth, so feel free to take a look at those.